Hello, in this video we will discuss about the glyoxylate cycle. So first of all we should need to understand about the introduction, and let's begin to understand, and next we will discuss about the whole process. So the glyoxylate cycle, a variation of the tricarboxylic acid cycle, is an anabolic pathway occurring in plants, bacteria, protists and fungi. The glyoxylate cycle centers on the conversion of acetyl-CoA to succinate for synthesis of carbohydrates. So the synthesis of the carbohydrate is important for the glyoxylate cycle. So first of all, we should need to understand about the cell of the plant as well as the cell of the prokaryotes bacteria. And fungi we will not understand in this video, but fungi is also involved in this mechanism. But let's begin to understand these things. So this is the plant cell. Contain nucleus. Nucleus contain chromosome, and that chromosome is in the form of histone protein and DNA. So the 40% DNA and 60% histone protein. Here is the mitochondria and chloroplast, and the important organelle is known as the glyoxysome. So the glyoxysome are present, and this is the eukaryotic plant cell, and it is also involved in the prokaryotic plant cell, but that is known as the blue green algae, but that is not involved in this mechanism too much and rare cases. But here is a vacuole, a mitochondria, nucleus and chloroplast. Chloroplast is used for the photosynthesis during the light reaction, but in this case the chloroplast is not involved in this mechanism. Why? Because during the seed germination, not produce chloroplast buried into the soil, and that means the chloroplast absence of the chloroplast, the sugar molecule development through a glyoxylate cycle is possible. So why it's important, the glucose, we will discuss also about that. And here is a bacterial cell. This bacterial cell is basically contain also glyoxysome, and here is you can see this plasma membrane, and this is the glyoxysome, orange color showing in the figure, while here is you can see that is a circular DNA as a chromosome are present in the center, while here the plasmid is the super smaller extracircular DNA, and it is cell wall. So the plasmid is also used for vector. So this is the prokaryotes, because not true nucleus are present. So let's begin to understand. Here is important organelle which that is present in the seed is known as oleoplast in seed. So this is the form of plastid, and this plastid is used for it, and because it is the storage part of the fat into the seed. So first of all, here is the sugar, which that is the character of the sugar is the anabolism and catabolism. So the anabolism can be synthesis of cellulose, NADPH, NADP plus and ADP. That is the important formation for the plant and animal. So here is the glycolysis and Krebs cycle is the important catabolic pathway, which that is the Krebs cycle gives the electron transport chain, and gluconeogenesis is the anabolic part. So here is the beta oxidation of fatty acid will be occur from the oleoplast go into the mitochondria. That fatty acid go into the mitochondria to beta oxidation of fatty acid to produce acetyl-CoA. So this acetyl-CoA production will be increased due to the deposition of the fat into the seed. So that seed is very important for the glyoxylate cycle. So this is the acetyl-CoA with combined water. So combined with the oxaloacetate to produce the citrate. So first of all, we should need to understand the Krebs cycle. First of all, we will understand about the Krebs cycle, and next we will discuss about the glyoxylate cycle. So first of all, here is you can see this is the coenzyme will be released, and the 2 carbon will be added in this compound oxaloacetate to convert citrate, and this citrate formation, is basically the 6 carbon containing molecule is looking in the figure. After the releasing of the water in the presence of aconitase to produce the cis aconitase, And after the addition of the again water in the presence of aconitase enzyme to produce isocitrate. And this isocitrate is very important character. This isocitrate will go into the glyoxysome, and after the go into the glyoxysome, the glyoxylate cycle will be run. So remember about that. Isocitrate is a precursor for the glyoxylate production. So the glyoxylate produced from the isocitrate when go into the glyoxysome of the organelle of the plant cell. So during the seedling time, during the embryogenesis of the plant. So after the isocitrate will produce the alpha ketoglutarate. First of all, we should need to understand the Krebs cycle. So this is the NADP plus convert into the NADPH in the presence of isocitrate dehydrogenase enzyme, and carbon dioxide will be released to produce alpha ketoglutarate. And this alpha ketoglutarate will produced in the presence of alpha ketoglutarate dehydrogenase to produce NADH, and carbon dioxide will be released to produce the succinyl CoA. Due to the addition of the CoA also during the alpha ketoglutarate dehydrogenation, so the succinyl CoA will convert in the presence of ADP, will convert into the ATP and release of the CoA enzyme. So the coenzyme. And after the releasing of the coenzyme, the succinate formation will be occur in the presence of succinate CoA synthetase enzyme. So the succinate synthesis of will be occur. And after this synthesis, succinate synthesis occurs. So the succinate dehydrogenase enzyme to produce FADH2 also from FAD plus, and to produce a fumarate. And this fumarate basically production to fumarase enzyme in the presence of water will produce the malate. And this malate is a 4 carbon containing compound. Due to the after the releasing of the carbon dioxide during the alpha ketoglutarate and the isocitrate will that the 2 carbon will be released, so the 4 carbon will be occur, and again produce the oxaloacetate. So this oxaloacetate will again run. But now we will discuss about important things you should need to understand. This is the oxaloacetate and this is the malate. Malate convert into the oxaloacetate. So let's begin to understand about the isocitrate conversion into the glyoxylate in the glyoxysome organelle. So this is the glyoxylate formation will be occur. So this isocitrate lyase enzyme used for it to produce the glyoxylate, and this glyoxylate is the malate synthetase enzyme to produce malate. After the releasing of the acetyl CoA addition, the acetyl group will be added and the coenzyme used to use for it. And in this way, in the presence of coenzyme and acetyl bind to produce again this glyoxylate 2 carbon containing to convert into the 4 carbon containing malate. So this malate convert again in the presence of malate dehydrogenase enzyme to produce oxaloacetate. This oxaloacetate can be go into the Krebs cycle again or go into another mechanism which that is produce the phosphoenol pyruvate. So suppose this is the 2 molecule oxaloacetate, and this 2 molecule convert in the presence of phosphoenol pyruvate carboxykinase enzyme in the presence of GTP to convert GDP and the NAD plus which that are used into the malate dehydrogenation, and the 2 phosphoenol pyruvate produce 2 molecule and 2 molecule of 3 phosphoglycerate produced, and this is a gluconeogenesis formation. So this mechanism is known as a gluconeogenesis, and the 3 phosphoglycerate molecule produce and ATP used to produce ADP, and phosphoglycerate kinase enzyme used to produce 2 molecule of 3 phosphoglycerate, and this NADH produced from the NAD plus. So NADH use to produce the fructose 1,6 bisphosphate. So the fructose 1,6 bisphosphate phosphatase enzyme used to release of the phosphate group to produce fructose 6 phosphate. It means the only one phosphate group add into the 6 carbon of the fructose. After this, isomerization will be occurred due to the isomerase enzyme to produce the fructose 6 phosphate to conversion into the glucose 6 phosphate. And after this glucose 6 phosphate production due to the isomerization to produce the mature glucose, this is the basically releasing of the phosphate. So the glucose 6 phosphatase enzyme to produce the glucose. This glucose produce the DNA replication, cellulose and starch formation. Cellulose mean the cell wall development, and ADP, ATP and NADH production. So what that means? It means the ADP or on the other hand NADPH and cellulose and starch will be not synthesis due to the absence of the chloroplast, and chlorophyll due to the seed will not produce the chlorophyll during the early stage of development. So the in plant the glyoxylate cycle occur in special peroxisome which are called glyoxysome. This cycle allows seed to use lipids as a source of energy to form the shoot during the germination. So the seed cannot produce a biomass using photosynthesis because of lack of an organ to perform this function. It means the chloroplast is not present. So that is why it is very important the glyoxylate cycle to produce the glucose for different mechanism and synthesis of the cell wall and cell wall and cell division due to the nucleotide production. So that is the important things you should need to understand. And in bacteria during endospore formation, during the unfavorable condition, the dormant stage will again activate to require the glucose. So the photosynthesis not occur due to the chloroplast is not present. So the early stage of the again reappearing of the bacterial cell required the glucose through a glyoxylate cycle. So the glyoxylate cycle is a very important for production of the glucose through gluconeogenesis. So this was the gluconeogenesis to produce glucose from the oxaloacetate. While the oxaloacetate will again can be go into the Krebs cycle to catabolism, but this was the anabolism from the oxaloacetate to produce the glucose. That is the anabolism of the